Yeah, thanks so much for the invitation to this great seminar here. It's a pleasure to be here and talk about a particular dynamical regime, which quite a bit of data that we analyzed over the past few years suggests that cortical networks seem to implement that regime. So, but before I dive into the topic, let me first show you the people that are behind the work that I'm going to talk about in the next 45 minutes. So it's based on a series of publications, which you find here at the bottom of the slide. So you can check out the details there, but also, of course, ask questions later on. I'm happy to answer any questions. So these works are a combination of experimental data analyses and theoretical work, experimental data from both macaque motor cortex and mouse visual cortex. So the macaque data was recorded at Aix-Marseille University in the group of Thomas Rocher, Alexa Riele, and Nicole Vogels. The mouse data comes from the Allen Institute in Seattle and was recorded around people like Tim Jarsky, Stephanie Seaman, Luc Campagnola, Shashan Jia, and Gabe Ocker was also involved in the study. Then from the theoretical side, especially the last paper, Stefano Recanatesi from the group of Eric Scher Brown and the University of Washington was heavily involved. And the first two papers are also largely large work together with people from Jülich, Moritz Helias, Markus Diesmann, Sonja Grün, Moritz Leier, Paulina Dabrowska, and Lukas Deutz. Okay, I guess everyone here in this virtual audience would probably agree with me that complex brain function really relies on the concerted dynamics of neurons in neural networks. And people have analyzed this coordination structure for quite some time now and try to understand correlations, which are the simplest measure of neuronal coordination. And over the past few years, the notion of neural manifolds became also more and more popular. So what are neural manifolds? Neural manifolds are basically portions of the n-dimensional space of neurons. So each axis here in the sketch basically denotes one neuron, and you have many of these in a high dimensional recording. And you can plot the activity of all these neurons as one point in this space. And what I mean with activity typically in this talk is binned spike trains. So you have parallel recordings of spike trains, which we bin in this case in larger and quite large bin sizes, since we're looking at coordination over long time scales. And then if you have a spike count vector for each bin, you can depict this as a dot in this n-dimensional space. And if you do this for many bins, then you end up with these clouds. And people have noticed in various works, and some of them are listed here at the bottom, that these clouds are not really isotropic across all directions in this n-dimensional space, but that activity is really constrained along certain directions due to coordination, due to interactions between neurons. And this gives rise to a decrease in dimensionality here from left to right. So one important question then is which mechanisms actually do shape this neuronal coordination? And one can study that question in with two complementary routes, let's say. So first, one could say a bunch of neurons most likely gets shared external input, which is correlated. And if it's strong enough, then it, of course, entrains local dynamics and thereby constrains the dynamics and reduces the dimensionality. Here in this talk, however, I would like to focus on the complementary view, which arises, or which is the component of coordination that arises even in the absence of any correlated or shared external input, just due to the fact that neurons are locally recurrently connected. So they're talking to each other and thereby coordinate their activities. And of course, understanding these intrinsic correlations in neural networks is also a topic that has been studied for many years and decades already. For example, reaching back to the famous work by Carl von Vreeswijk and Haimson Polinsky on the balanced state. So typically one studies certain structures in this connectivity, and the most simple example is to study excitation and inhibition. And what people found out in this study, and also later in the influential works by Alfonso Renard and Tom Tetzlaff and others, that if there's more inhibition than excitation in these networks, then one ends up in this balanced state with low average correlations. And one can, of course, make this picture more complicated, add more and more population structures, but also more recently, following work by Mastro Jeppe and Ossilch, people looked at other types of low rank structures that might possibly come about by learning or training a neural network on a particular task. And here in my talk, I would again change the viewpoint slightly, going away from these deterministic network structures and looking at second order connectivity statistics, so variability in connections, because we know that this is ubiquitous in cortical networks. So there's the classical paper by Song et al., for example, that studied distributions of synaptic amplitudes that vary over multiple orders of magnitude, and also more recently, larger scale connectivity analysis, for example, from the Allen Institute, shows that there's many heterogeneous features of connectivity, for example, sparsity, and also synaptic amplitudes, of course, and others that contribute to that variability in connections. So why is variability in connectivity important? Well, one can simply imagine that variability in structure basically leads to variability in dynamics. And to showcase this here, I have 3 simulations of inhibitory networks of leaky integrated fire neurons. And just by eye, you see in the raster plots here on the left that all of these 3 networks operate in the balanced regime because we see this asynchronous irregular activity. However, if we now look at covariance distributions, and I talk about spike count covariance distributions again here across long time scales, then we see striking differences between these 3 different scenarios, namely, well, first of all, the mean of the covariances is always close to 0. That's basically the balanced state. But the width of the distribution is pretty different across the different scenarios. Here at the top, where variability in connectivity is low, the variability in coordination and in correlations is also low, while at the bottom you see a very different picture, namely neuron pairs that are pretty strongly correlated and others that are pretty strongly anti-correlated. So why is this variability in covariances important? Well, let's go back to the simple sketch of these neural manifolds. So one can try to characterize the dimensionality of neural activity using various methods. And one simple method is to calculate the so-called participation ratio, which is a quantity that is based on the covariance matrix of neural activity and typically expressed in terms of the eigenvalues of the covariance matrix lambda I here. But one can also rewrite this in terms of the statistics of the covariance matrix. And n here is the number of recorded neurons. And if n is large, you see that m and s here are the main determinants of the dimensionality. And what is m? M is the mean of the cross-covariance distribution, which is low in the balanced state, as I just argued and showed in the simulations. And s is the width of this distribution here, which, as I showed you, can be very large. So in the balanced state, we would assume that it's basically this variability in covariances that most strongly constrains the dynamics and the dimensionality of these systems. So how does this look like actually in real data? So in the first part of the talk here, I show data from Marseille recorded in macaque motor cortex using a UTE array, which is a 10 by 10 electrode arrays spanning 4 by 4 millimeters of cortical surface. And this allows us to record parallel neuron activity of up to 150 neurons, as you see here in the raster plot on the right. And this data was recorded during a reach to grass task of the monkey, but we particularly chose those periods of the task where the monkey was actually sitting still. So there's no movement related transients in the activity. So you see here, the raster plot looks rather boring, asynchronous, irregular, stationary activity. So if you now calculate spike counts across these 400 milliseconds here and correlated these spike counts across different trials, then you would end up with a covariance distribution which looks like this, which resembles what I've shown you in the simulation, namely that the mean of this distribution is pretty close to 0. But we see neuron pairs that are pretty strongly correlated and other neuron pairs that are pretty strongly anti-correlated. So there's large variability. And the question now is, what does this large variability actually tell us about the underlying network and its other dynamical features? So to study that question, unfortunately, the typical population level modeling approach that we use that basically groups together statistically similar neurons, for example, excitatory neurons and inhibitory neurons into populations and studies connectivity on an average level across these populations, which is a powerful technique to boil down the dimensionality of the problem. But unfortunately, it's only yielding population averaged dynamics, so mean firing rates of the populations and mean correlations. And as I showed you, we want to go beyond this mean because we want to study also this dispersion, this variability in coordination structure and correlations. So we need to go to the level of individual neurons and individual synapses, basically, to study that topic. And I started doing this in 2016 in this paper here in Physical Review X, where I studied very simple neuron networks, namely networks of binary neurons that are here also depicted as spins that can either point upwards or downwards, so akin to magnetic materials, basically. But here we also studied different populations like excitatory and inhibitory neurons, directed connectivity, and so on. But still, the neuron dynamics was very simple. It was either on or off dynamics of the single neuron. And what we showed in the study was that using some Gaussian approximation of the input, the correlation structure or covariance structure of this binary network can actually be mapped to the correlation structure of a more simple network, namely a linear rate network of Ornstein-Uhlenbeck processes, basically. And what other people showed is that basically, if you start from a spiking leaky integrated fire network, you can do the same mapping and end up with a linear rate model. And if you look at very long time scale correlations, then actually you get basically model independent relation between structure and dynamics. So between the covariance matrix C and the effective connectivity matrix W. I'm saying effective connectivity because in this mapping here, one performs some kind of linearization around the working point. So the working point itself enters in the effective weights, which we have here in this connectivity matrix W. And by the way, this matrix D also stems from the operational point, so basically from the firing rates of the neurons. So from a theoretical point of view, one could say, well, this is great because now we have a relation that tells us exactly if we have individual connections, what are the individual covariances in our network. And people studied this equation here in various flavors a lot. And here you see a few of the works that basically derived this equation, and starting with a linear response theory work by Benjamin Lindner. And from a theoretical point of view, it's really a nice formula. But when it comes to application to experimental data, then we face a severe problem. Namely, that this is a matrix equation of n by n matrices, where n is the network size. And even in the most modern electrophysiological recordings or other types of activity recordings, we can still only see a very small fraction of neuron activities. So here indicated by this green square. So the rest of the matrix is basically unknown to us. So this means that we cannot simply try to invert this relation, for example, and infer individual connections from measured covariances, because there's basically an ill-conditioned problem. However, as I said, even the Utah array recordings nowadays give us neuron activities of 150 neurons in parallel. And there's, of course, other techniques that might even give us more neurons. So this means that this little green square here actually has 150 squared values of covariances. So there's basically enough data to do some proper statistical analysis. So what this means is that rather than having a relation between individual covariances and individual connections in the network, what would be better to have in terms of applicability to the data would be a relation between the statistics of these covariances and the statistics of these connections. So then one could try to invert the problem and infer properties of the underlying network connectivity from the measured covariance statistics. So how do we get to that point? Well, simply averaging basically this equation here is not super feasible, because this -1 here actually denotes in matrix inverse. So this connectivity matrix enters here as an inverse matrix. So in certain scenarios, one can try to expand this inverse matrix in a geometric series, but then we end up with infinitely many powers of our connectivity matrix W, and this is also hard to average. So we need to find some other kind of language in order to be able to perform some systematic averaging of these types of equations to bring in the connectivity statistics rather than the individual connections. So in this different language is statistical field theory. And how does this go here for this particular problem? So first of all, let me remind you that this equation here is pretty much model independent. It doesn't really depend on whether we started with a binary network or leak integrated fire network or rate network. In certain dynamical regimes, they all boil down to this equation. This, however, means also that we can choose a particularly simple model that produces this relation between structure and dynamics. In our case, this is the linear rate model, so coupled Ornstein-Uhlenbeck processes. So xi here is the fluctuation of some neural activity I. So there's a leak term, there's an effective connectivity matrix wij, and then some uncorrelated Gaussian white noise input that drives these fluctuations here. And just with a few lines of algebra, one can go from this dynamical system to this relation between structure and dynamics. But one could now say, well, why do we do this? Because now we went one step back. We already had the relation between structure and dynamics, and now we go back to the dynamical system. So why is this helpful? Well, because the dynamical system, so stochastic differential equations, we can again translate into an object which is called a path integral, P of x of t, which is the probability of observing a neural trajectory x of t over time of this dynamical system here. And this path integral is basically defined by an object which is called the action S, which enters here in the exponent. And the action S encodes basically the whole information about the neural network. And you see the action is quadratic in x and x tilde. X tilde is just a helping field that lets us express the dynamics in such a form. So, but this means also that basically if we integrate everything out here, then this is just a Gaussian integral, so easy to solve. And this basically stems from the fact that we started with a simple linear rate model. And you see parts of the dynamical equation also here in the action. You see the differential operator here, you see the one from the leak term, you see the effective connectivity matrix, and you see a scalar D, which is basically the strength of the Gaussian white noise that we integrated out. So having this object here is very helpful because this language here is being used in many different disciplines in physics, and many different methods have been devised over the past few decades to deal with these objects. And in particular for our problem now, it's a convenient object because the connectivity now enters linearly in S and S enters in an exponent. So what we have to average is an exponential of something that is linear in W. And this is a well-controllable object. It's a characteristic function. And we can basically expand this incumulance of the connectivity. So the systematic averaging of the connectivity statistics P of W is now possible in this formulation. And in this book here, you find basically details in how you go from the dynamical system to these disorder averaged path integrals. And of course, I can also go into details in the questions later on, but now let me give you basically the gist of what happens next and do the theory in a nutshell. Basically, if you average over the connectivity statistics P of W, then you end up with an action that is not quadratic in axis, so an integral which is not a Gaussian. So we cannot solve this analytically, and we have to resort to approximation schemes. And these approximation schemes are well studied in other fields in physics, so we can basically employ the methods that have been used there. And in a nutshell, basically the simplest type of approximation is called a mean field approximation again, which at the end of the road basically yields again mean covariances. But as I said, mean covariances here for this data are not so much informative because it doesn't tell us about what's going on for individual pairs. So we would rather like to have more information on the dispersion here of these covariances in the data. And to study these, we need to go beyond mean field and study fluctuations, which then in the end give you the variance of covariances, so the width of this distribution. And in a nutshell, what we get is basically that this width of the distribution delta here is determined by a property which we call recurrency R. So this is a property of the effective connectivity. So we can plot the effective connectivity in terms of its eigenvalues here in the complex plane. So the x-axis is the real part of the eigenvalue, the imaginary part of the eigenvalue is the y-axis, and each dot here is one eigenvalue. And what R now is the spectral radius of this bulk of eigenvalues. So basically, it also boils down to the largest real part of the eigenvalues which we have here. So this is a summary measure of connectivity statistics. And in principle now, since we have this relation between the connectivity statistics and the width of the covariance distribution, we can measure the width of the covariance distribution in an experiment, plug this in here and infer the radius R. And what we get is that the radius R is close to 1. And why is 1 important here? So you see this dashed vertical line here at 1. So remember, we're dealing with linearized dynamics here, and the linearized dynamics become unstable at a certain point. And this is exactly if one eigenvalue has a real part which is larger than 1. So being close to 1 means that we are close to the point of linear instability, and this is what we call the strongly recurrent regime. And in the remainder of this talk, I would like to show you features of this strongly recurrent regime that the theory predicts and that we then investigated in data. So the first feature was the diverse correlations, so the broad width of the distribution of correlations, which I already talked about, and which helped us to infer basically this dynamical regime. And the next feature would be a rich dynamical repertoire. So to understand this, we go back to the eigenvalue spectrum here. Again, you see the plot that I showed you on the previous slides. So each of these eigenvalues here also comes with an eigenvector. So for example, this eigenvalue here, which is a population eigenvalue that is determined by the overall excitation inhibition level in the network, and this is a balanced network, so this eigenvalue is strongly negative here. So this eigenvalue let's say in a purely inhibitory network where we don't even have recurrent excitation, this eigenvalue has entries, this eigenvector has entries which are uniform across all neurons. So this means that if we plot this in the n-dimensional space, this is a vector that somehow shows in some kind of diagonal direction. And all these other eigenvalues also are associated to eigenvectors, but since these eigenvalues are generated from the randomness, from the variability in connectivity, also the eigenvectors look much more unstructured. So they point in some different direction in the n-dimensional space. So why are these directions important? Well, we can now imagine to perturb the system exactly in this direction or in that direction. So basically excite neurons or inhibit other neurons in a particular way so that we excite dynamics along that particular direction in the n-dimensional space. Then we will also see very stereotypic response. So how do we get this for the linear model? So we have the coupled Ornstein-Uhlenbeck processes here. So we can just do an eigenvalue decomposition of W here by multiplying this equation with some eigenvector. So then we basically end up with a decoupled set of equations for so-called eigenmodes, V alpha here. So they're decoupled because we do not have a connectivity matrix here, but just an eigenvalue. And these are one-dimensional linear equations, which we can simply solve. And what we get are exponentials. And the time constant of this exponential is the neuronal time constant, tau, but divided by 1 minus lambda alpha. So the eigenvalues of the connectivity determine the timescales of these eigenmodes. And remember, in the strongly recurrent regime, we have eigenvalues which are close to 1. So if we plug in a 1 here, or something that is close to 1, let's say, then we see that this effective time constant here becomes very large. Yeah, so this means that if we kick the system exactly in this direction that corresponds to this most unstable eigenvalue here, then we see a very long response. So the system will remember for a long time that it has been kicked. And also the autocorrelation function shows a decay, which is much slower than the decay of individual neurons. If we do the same thing for this outlier eigenvalue here, so for the population eigenvalue, then, so since this is a very negative in this case here, we get an effective time constant which is much smaller than the neuronal time constant. So the system will almost immediately forget that it has been kicked. So this is a so-called fast tracking property that have been analyzed by many people already quite a few years ago. So the autocorrelation also drops off very quickly. And the good thing about this regime here is that we also have the whole bulk in between. So that means that we also have a whole repertoire of timescales in between these two extremes, including oscillatory modes if we have some imaginary component here of the eigenvalues. So this is the rich repertoire of timescales. So the problem with this repertoire is that it's really defined by the connectivity. And since we do not know the connectivity in an experiment, again, so we cannot really see this directly. We do not know how to project our activity basically in order to see these individual eigenmodes. But what we can do is a simple principal component analysis of our data. And we did this here for model data. So you get these plots of variance explained across PC dimension. And you can study again the loadings of the eigenvectors corresponding to the different principal components. And you see the loadings are also pretty much unstructured here, similar to the unstructured loadings that we saw for the connectivity eigenvectors. And then you can also project your activity onto these loadings and calculate autocorrelations or autocovariance functions. And you see the differences in timescales here again. And these timescales are not the same as the timescales of the connectivity eigenmodes, but they are non-trivially related. And this means, so for example, if we wouldn't have this whole repertoire of timescales, which we inferred from the connectivity, we couldn't also have this large repertoire, which we see here in the principal component spectrum. So the advantage of this method is that we can also just do this for the data here for macaque motor cortex again, and we see a similar picture as in the model. So the principal component eigenvectors look pretty much random, and the autocovariances of the individual projections show various different timescales. So ignore the peak here at 0, which is due to the spiking nature of the signal here, which is of course not here in the linearized model. So this gives us possibilities to basically test these predictions using standard principal component analyses. So that was the rich dynamical repertoire which we have in the strongly recurrent regime. Next, I would like to talk about coordination, long-range coordination that can come about even if we do not have long-range connectivity, but only short-range connectivity. And to motivate this, I would like you to focus again on the figure that I just showed you and focus here on this loadings of the first principal component. If I would have plotted many more loadings here, you would have seen that it's not only a tiny fraction of neurons that really contribute to this first principal component, to this mode of activity, but it's really all of them, some more, some less. But the question is, how can all of these neurons basically coordinate their activities to be part in the same eigenmode? How do they do this? And in particular, we asked this question because this data comes from a Utah array recording where different electrodes are actually 400 micrometers apart from each other. So here again, we have the 4 by 4 millimeters that the electrode array spans. And basically in each of these little squares, we have one electrode. We can now plot markers where basically the neurons have been recorded. So this is the rough position of the neurons. Each triangle is a pyramidal cell, each putative excitatory cell, each circle is a putative inhibitory neuron. And now we can pick one of these neurons as a reference neuron. So in this case here, it was an excitatory neuron. And then color code in the markers, the correlation of all the other neurons with respect to this one neuron here. And what we see is basically a salt and pepper pattern all over the place, all over the Utah array. So it's not that basically strong correlation is only happening in the close vicinity here of this neuron here indicated by this red circle, which is a few hundred microns. So basically the range of direct axonal arborization of these neurons. But we see strong positive and negative correlations basically also at very large distances. For example, in particular here, these neurons are far away from this neuron. This is a few millimeters here. And still you see a pretty strong anti-correlation of this neuron with this neuron, for example. So one simple explanation, of course, would be to say, well, there is a direct connection from here to there, which enables these neurons to talk to each other. But if we focus more closely here, this is like a magenta color, which is a negative correlation. Yeah? So two excitatory neurons, since they excite each other, we would expect that the correlation is positive. And also shared external input of these two neurons would most likely yield a positive correlation. So the question is, how does this negative correlation between excitatory neurons come about? And the answer is, well, we should not only study direct connectivity between neurons, but also all these indirect pathways through the network where potentially inhibitory neurons are somewhere in between that basically reverse the positive effect that this excitatory neuron has on its postsynaptic target. And if we sum up all of these pathways, we might effectively get negative correlations between excitatory neurons. And just thinking this one step further, if we're not restricted to the range of direct connections, but have indirect pathways, then of course we can also create long-range correlations in our network. So the question then is, when do these indirect interactions actually matter in our networks? And to study that question, we focus again on this central equation which we started from. So the central the equation which links the covariances and the connectivity. And I said this is an inverse matrix which we can expand in a geometric series. So what are these powers w to the power n? What are these here? So well, w to the power 1 is a direct connection between two neurons. W to the power 2 is an indirect connection via one intermediate neuron. W to the power 3 has two intermediate neurons, and so on. So these are the indirect pathways that I was talking about just before. And now remember again, in the strongly recurrent regime, we have eigenvalues which are close to 1. So let's say 0.95 or something like this. And if we take 0.95 to some power, let's say power 5 or so, this is still a sizable number. So this means that these indirect connections in this regime still matter. So, and since we see these negative correlations, it seems that in the strongly recurrent regime, actually these indirect pathways are the dominant contributions to the interactions between two neurons. And we checked this, of course, again in a model where we distributed excitatory and inhibitory cells on a two-dimensional sheet with periodic boundary conditions, and we implemented some sparse and distance-dependent connectivity. So a connection profile that falls off exponentially with distance, with some reasonable time scale, length scale, sorry. And then did the field theory for these models. So basically we encoded in the second order and first order statistics of W, we encoded this distance dependent, and then calculated how the correlations will fall off with distance and how this is related to the time, to the length scale of the connectivity. And this is what comes out. So first let me mention, so we're looking again still at the variance of covariance, so the width of the distribution, how this falls off with distance here on the x-axis. And the different lines here are different recurrencies. And you see that the fall off becomes much more shallow the stronger we get into this strongly recurrent regime. Furthermore, we could predict a certain ordering of these variances, of these widths for excitatory and inhibitory connections or pairs of neurons, and also the EI pairs in between. And this we could then check in the experiment because we know where the neurons were roughly located. So we can basically look at covariances in a distance resolved manner. And this is what we get here. So you see again the logarithmic axis here on the y scale as in the theory. So a linear decay here denotes an exponential decay on a linear scale. So on the x-axis you see again distance here now in millimeters, and this is an exponential fit here. And the length constants are in the millimeter regime, so far above the few hundred microns that we would assume that local axonal aberrations would make up. So this tells us that through this indirect connectivity, we can actually reach much further, and this seems to explain also the salt and pepper pattern and the long-range patterns that we see in the data in macaque motor cortex. So the next dynamical feature of the strongly recurrent regime would be the fact that coordination patterns are not really static and set in stone, but they're highly dynamic. So let me motivate this. So in this relation that I showed you between covariances and connectivity, we were talking about effective connectivity. I said, since we are dealing with linearization processes, the operating point around which we linearize enters basically the effective connectivity matrix. This also means that if we change now the operating point, let's say the firing rate of the neurons, by providing a different level of external input as sketched here, then this means that we also changed the working point and thereby the effective connectivity. And if the effective connectivity changes, then the coordination patterns will change. So here you see one network, basically. This is the anatomical connectivity here. And then we have two states, the dark blue one and the light blue one. And what we've done is we provide a certain level of external input in each state, and it is different between the two states. So that means, for example, the gain of this neuron here is low in the first state and high in the second state, because the firing rate will be higher in the second state. So this means that we end up, so one and the same anatomical connectivity will end up in two different effective connectivities. So how did we do this in practice? So we said these input levels that neurons receive are drawn from a Gaussian distribution here. And we choose actually the same Gaussian distribution for the two different states here, N1 and N2. But what we do is we swapped the neuron identities. So basically in one state, neuron 1 gets a high input, and then in the other state, it's neuron 2 that gets the same high input. And then they change basically over the states. And they do not change too much, because if we calculate the correlation coefficient between these two inputs, then it's still quite large, 0.9. And if we look at the statistics of firing rates and covariances, we do not see any difference, because it's a large network, it's self-averaging. So it doesn't really matter whether I change some neural identities or reshuffle some inputs or so. The overall firing rate distribution and covariance distribution is still the same. But what changes is the individual firing rates and individual covariances. So neurons basically move the position within these distributions. So for example, if in state 1, there was a neuron that had a high firing rate here, in state 2, it will have a lower firing rate. And you see that the correlation of the firing rates is smaller than the correlation of the input levels. But what is, again, much smaller is the correlation between the covariance patterns in the two different states. It's only 0.4. And we also quantified this a bit more thoroughly. So basically, we measured multiple times in the same state and then calculated the correlation coefficient within the same state of the firing rates and of the correlations. And this is close to 1. This tells us that we measured well enough and have enough data to estimate firing rates and covariances properly. So we do not have much noise in this data. But if we do the same thing between different or across two different states, then we see that the correlation coefficient significantly drops, more so for the covariances than for the firing rates. And if we do this, play this game now for different networks that have different recurrencies here on the x-axis, we see that the firing rates, the correlations stay quite high all the time, but the covariances actually, the correlation coefficient of the covariances goes down very strongly, especially in the strongly recurrent regime. We would expect that coordination patterns strongly change. So we can now test this in the experiment again, and this is the same experiment that I was talking about before. So we had a monkey that did a reach to grasp task. But as I said, we're always looking, since the linearization process is involved, we're always looking at data where the monkey is sitting still, where there are no large transients of activity. However, there are multiple of these epochs in the task where the monkey is sitting still. So there's the first epoch here at the trial start, S1, or called S, basically where the monkey didn't get any cue and doesn't know basically which type of grip it has to perform or what kind of power force it has to apply. So there's basically no knowledge. And then there's a second period where the monkey is sitting still. It's a preparatory period where there was already a cue. So the monkey knows which grip type it has to use. So it should be in a different behavioral state, right? It has more information than before. The distributions of firing rates and covariances of these two different epochs in the task are, again, very similar. However, if we look at the individual covariance patterns, and here you see, again, one of these salt and pepper plots, this time it's an inhibitory reference neuron, you see that the colors here basically change from S to P. completely, right? So for example, here, this neuron was strongly anticorrelated in period S, while it's positively correlated here in P. And again, we did this more thoroughly. So we split each epoch into 2 parts and then measured basically what is the within state correlation coefficient and compared this to the across state correlation coefficient. And again, for the firing rates, we have enough data. So the within state correlation coefficient is close to 1, while the across state rate correlation coefficient drops significantly. This is basically the reshuffling of firing rates within the distribution that happens from this state to the other state. And more importantly, for the covariances again, we also did the same thing. We calculated the correlation coefficient. The within state correlation coefficient now is also a bit lower because there is not so much data here. So there's a bit of noise in the estimation of correlations here. So this basically moves down these grayish points here. But more strikingly, the across state correlation coefficients go down much more. And this shows us that these covariance patterns really strongly change and can be modulated by the system. So this modulation and control of covariance patterns is also the topic of the last feature, where we turn the view to dimensionality again, which I started with. And I show that in the strongly recurrent regime, actually dimensionality is not only relatively low, but also strongly tunable or controllable. So again, let me remind you of this very simplistic plot here of this manifolds. From left to right, we see a decrease in dimensionality. And now if we employ, for example, the participation ratio, then I showed you that what determines this dimensionality is the covariance statistics. And in particular, if the distribution of covariances becomes broader and wider, then the dimensionality goes down. And then I showed you that the distribution of covariances gets broader, the more recurrency we have in the network. So the larger the spectral radius of the bulk of eigenvalues becomes. So in total, we get a relation between the recurrency R and the dimensionality of the global activity. And we tested this again and calculated this using our field theoretical methods. So this is basically this dashed gray line here, and tested this in models. So the rate model basically follows the prediction. And then we also tested this in spiking, leaking integrated and fire networks. These are the darker colors here. And also tested this across various network topologies. So purely inhibitory network, excitatory inhibitory networks, some with spatial connectivity profile. And consistently, the dimensionality, the relative dimensionality, so divided by the number of neurons, goes down when the recurrency goes up. And with relative dimensionality, so large recurrency, let's say 0.9 or so here, doesn't mean that dimensionality ends up at being 1 or 2 or 3 or so, right? But it's like a tiny fraction of the possible degrees of freedom that a network would have. What is also important here is that if we study how the dimensionality now changes when we change the recurrency and we look at relative changes, basically, then we see that the relative change is largest in the strongly recurrent regime. So this means if we are here somewhere in the strongly recurrent regime, we wouldn't expect that dimensionality is really some fixed number or so, but we would expect it to change across time quite strongly, as we saw for the individual covariances of the network. And to test that, we went to data from the Allen Institute, so neuropixels recording from mouse visual cortex during spontaneous activities. So this is the Allen Brain Observatory data set. Here you see an example raster plot of some activity, spiking activity of neurons. And by eye, due to the sorting that we employed, you see that there are strong transients in activity here. And these transients are related to the running speed of the mouse because the mouse was free to run. So when the speed goes up, we see also these firing rate changes. So from a theoretical point of view, these non-stationarities are, of course, harder to handle, but they also provide us now with a tool to study how dimensionality and coordination change. Because what we could do is employ a hidden Markov model analysis and basically segment these data here into individual states. And you see some of these states are clearly associated to running, for example. And then we studied how dimensionality is made up within these states where we have more stationarity, and also how it changes across states. And also, by the way, we also perform some control analysis where we perform some low rank, so some latent factor analysis to remove potentially external input driven low rank activity components. But what it boils down to is basically plots like this, where we have again the dimensionality change here on the y-axis. And on the x-axis here, you see different areas of the visual hierarchy. And you see that dimensionality change is, as predicted by this strongly recurrent regime, is very large. So on the order of 50% or even more, and much larger than the changes within individual segments of the same state. So this was the strongly tunable and controllable dimensionality that can be modulated by changes in the recurrency. And these changes in recurrency can come about by changing of the operating point, as I said. So I've talked a lot about recurrency. And of course, I'm not the first one to study that summary measure of second order connectivity statistics, so the spectral radius. And the studies go back already to the 1980s, where people like Hans-Jon Polinsky and Hans-Jürgen Sommers and others and so on studied how this spectral radius, how this variance of connections is affected by synaptic strength or symmetry in connections. Also, people studied what happens when you introduce multiple populations, sparseness of connectivity, different neuronal gains, but also spatial effects, as I showed you. And here in the last few minutes, I would like to show you yet another feature, which is a very microscopic feature of neuronal connectivity, namely network motifs between triplets, tuples or triplets of cells. So reciprocal motifs, convergent motifs, divergent motifs, and chain motifs. And I show you that these actually scale up to determine the recurrency and thereby the dimensionality, or the global dimensionality of neural activity. So using field theoretic methods again, we calculated a correction term. So where basically these other features here made up a recurrency, which we call sigma here, and we calculated some modifier R motifs of this quantity. And then we went to the experiment again and tried to analyze, well, how abundant are these motifs actually? And the data from the Allen Institute, this is multi-patch recordings of up to 8 cells in parallel, actually published here this data and the manuscript in 2022. We analyzed this data specifically for the abundance of these different types of motifs. And what we found is that most of them are actually overrepresented in visual cortex, and we could plug in the numbers into our formula for the radius modifier, the recurrency modifier, and found that the motifs actually tend to reduce the recurrency, so make the network a bit more stable. But one should be careful by interpreting this number, because it's just a snapshot. Because as I said, effective connectivity changes with the operating point. And if we now have different populations in the network, and some are maybe in one state down-regulated and others are up-regulated, this also means that their motif contribution to the recurrency changes. So really the motifs form some kind of structural scaffold that can be differentially engaged by the different activation levels of subpopulations, and thereby recurrencies and dimensionalities could also change across states. So to summarize, I showed you that our data from macaque motor cortex and mouse visual cortex seem to suggest that these networks operate in the so-called strongly recurrent regime with a large spectral radius of the bulk connectivity eigenvalues. And this dynamical regime has a few very interesting properties, namely that correlations are very diverse and also these cell and pepper patterns. We have this rich dynamical repertoire, many different time scales in the system that could be excited and used, for example, for memory, the longer ones, or for fast tracking, the shorter ones. The networks, the neurons are able to communicate to each other effectively over larger distances, even though they are not directly connected. And coordination patterns in general are highly dynamic and can be tuned, and also the dimensionality can be tuned, and it's relatively low, so relative to the number of neurons in the network. And the origin of all of these features is really the variability and the patterns of connectivity. So if we focus on pairs of neurons, there's multiple pathways in the network that these could interact with each other. And there's much variability basically in between here that effectively generates all of these dynamical features. So with this, I would like to thank my collaborators and my funding for these projects, and thank you for your attention. And I'm of course happy to answer any questions, and feel free to also check out these 3 publications where you find more details on these topics.